Okay, let's get started. Good morning, everyone. Good morning and good afternoon to those joining us abroad. My name is Cassie Maxman from the IQ Secretariat. Thank you for joining us for today's webinar, Low Clearance Drugs, Long Incubations, a comparative evaluation of long-term hepatocyte models for enzyme fraction metabolized, presented by the Quantitative Fraction Metabolized Working Group in coordination with the Translational and ADME Sciences Leadership Group. We are pleased to welcome you to this webinar sponsored by the IQ Consortium. The IQ Consortium was established in 2010 as a technically focused, not-for-profit organization comprised of pharmaceutical and biotechnology companies. Our vision is to advance innovative solutions to biomedical problems and enable pharmaceutical companies to bring quality medicines to patients. Our mission is to advance science and technology to augment the capability of member companies to bring transformational solutions that benefit patients, regulators, and the R&D community. Before we introduce our speakers, a couple of housekeeping items. All attendees will remain muted. For today's panel, the chat function has also been disabled. However, you may submit questions during and after the presentation using the Q&A panel found at the bottom of your screen along your toolbar by selecting the More tab. Questions will be addressed by our presenters at the end of the presentation, time permitting. Following this webinar, for inquiries regarding membership or this presentation, we welcome you to contact the IQ Secretariat. You may also view a recording of this presentation and past webinars by visiting our IQ Consortium YouTube channel. Thank you. I will now hand it off to our moderator, Angela Doran, with Pfizer to introduce today's speakers.
Thank you, Cassie. Confirming that you can hear me.
Yes, we can hear you.
Perfect. Thank you. Thanks, Cassie. Thanks, everyone, for joining today. As Cassie mentioned, I'm Angela Doran, Senior Principal Scientist in the Pharmacokinetics, Dynamics, and Metabolism Group at Pfizer. And along with Naresh Hariprasad, I co-lead the Quantitative Fraction Metabolized Working Group. I'll be moderating today's webinar. Our presentation today summarizes a collaborative multi-company effort of this working group to better understand the opportunities and the challenges associated with using long-term hepatocyte systems for predicting enzyme fraction metabolized of low clearance compounds. But before I begin, we'd like to recognize the many current and former members of the working group whose expertise, along with data generation, scientific discussions, and persistence over several years of this working group have made this work possible. It's truly been a team effort, and we're really grateful for everyone's contributions and to the IQ. Next slide. Our speakers today, we're fortunate enough to have three distinguished scientists from the working group to present. Together, they represent the breadth of the expertise that has shaped our effort from mechanistic understanding through the experimental design and through data interpretation and application. Mrs. Helen Tracy is Associate Director of PBPK Modeling at GSK and brings extensive expertise in mechanistic ADME, drug-drug interactions, and model-informed drug development. Dr. Ting Wang is a Senior Principal Scientist in DMPK at Boehringer Ingelheim and with deep expertise in vitro metabolism at ADME, enzyme phenotyping, and bioanalysis. And Dr. Chuck Locuson is a Senior Director of DMPK at Agios Pharmaceuticals and has broad experience leading drug metabolism and pharmacokinetic strategies across discovery and development programs. Next slide. So really broadly, our presentation is organized into three sections that reflect the progression of the working group's efforts, beginning with study design to method development and practical application. We'll begin with an overview of the study rationale and the experimental framework, followed by an evaluation and optimization of the chemical inhibitors, and conclude with the application of the approaches to fraction metabolized determinations using representative case studies. I appreciate you all joining us today. We hope you find the presentation informative and useful. With that, I'll hand it over to Helen to get us started.
Thanks, Angela. As Angela said, I'm going to give an overview of the working group, and this slide highlights why the group was formed and our objectives. So to assess object DDI or drug interactions, we need to be able to accurately quantify the fraction metabolized of small molecule drug candidates by drug metabolizing enzymes or DMEs. And traditionally, this has been achieved preclinically by conducting reaction phenotyping assays in vitro systems such as human liver microsomes or hepatocytes with chemical inhibition. And these assays are often supplemented with incubations with recombinant enzymes. And all these in vitro systems are have short incubation times. But increasingly, small molecule drug candidates are metabolically stable, low turnover compounds, and these short-term incubation systems aren't suitable for detecting their slower metabolism. And this often results in inaccurate measurement of clearance rates and subsequent DDI risk. So long-term hepatocyte systems for measuring clearance and supporting phenotyping of low turnover compounds have been available for a number of years, but they're not well established for DME phenotyping. And this results in a lack of confidence in FM estimates for low clearance compounds. So some of the key gaps for phenotyping of low clearance compounds include a lack of systemic cross-platform and cross-lab benchmarking of these systems, resulting in questions around system performance and reproducibility, and uncertainties around model selection. There's also a limited amount of data on the selectivity and stability of chemical inhibitors for use in these long-term under long-term culture conditions, despite their established use in short-term incubations. And these gaps led to the formation of this working group. And our objectives were to conduct a systematic evaluation of long-term hepatocyte systems for predicting FM for low clearance drugs, to improve predictive practices and help inform the scientific community and regulators. So we were comparing across systems rather than optimizing each system in isolation to help address some of these gaps. Next slide, please. So this slide gives an overview of the timeline and effort of the working group and some of the key challenges we faced. Our work was split into 3 major blocks, which are highlighted down at the bottom of the slides. We kicked off in 2021, and during the first couple of years, we conducted literature-based scoping exercise to select our hepatocyte systems, prioritize which enzymes we wanted to look at, and then curate evidence on substrates or potential substrates and inhibitors suitable for these enzymes suitable for long-term culture. And this information was captured in a shared database. We initially planned to evaluate 2 tiers of enzymes, tier 1 covering the major CYPs and tier 2 covering the major non-CYP drug metabolizing enzymes. However, as we approached the experimental phase, where we recognized that this was perhaps overly ambitious given the available resources, so we narrowed our focus down to the major CYP enzymes, or just the major CYP enzymes. We began experimental work in 2023 and continued over the next couple of years. And a key setback or issue that we faced was with one of our case study drugs, repaglinide, where we found that the metabolite attributed to CYP2C8 in the literature wasn't the one formed by that enzyme in our working group labs. And this caused a lot of head scratching and sample reanalysis, and ultimately to delays in completing the experimental phase. But we did eventually get there, and we've spent the last year or so analysing the data and tying it all together for communication in a manuscript. Next slide, please. So this slide breaks down our process in finer detail, starting with the foundational study elements, which are highlighted in blue. So first we had to decide what assays or models we wanted to look at. Did we want to look at cells versus tissues, suspension versus plated hepatocytes, co-cultured systems, or anything else? And once we sort of settled on this, we had to think about what enzymes we want to look at. And as I mentioned earlier, we settled on looking at the major CYPs. And then once we decided on our enzymes, we moved to our probe substrates. Ideally, these would be low clearance compounds, and where possible, they were. But one major challenge that we faced was the limited number of low clearance compounds, well characterized for FM, that were found in the literature, could be found in the literature. Our next step was to perform an evaluation of the potential inhibitors in our selected hepatocyte models. This is highlighted in brown on the slide. Ting is going to discuss these in more detail, these in more detail later. So I'm just going to give a brief outline of our processes. We started with a literature-based search for potential inhibitors based on their mechanism, potency, and selectivity. And then we conducted an assessment of the suitability of potential inhibitors for use in long-term cultures. And this was based both on a literature search, on literature data, and experimental data generated within the working group. Our final step was to evaluate our selected inhibitors in the hepatocyte systems to determine appropriate concentrations to use in the estimation of FM. Our final step was to then use our selected substrates and inhibitors to determine the FM of 2 well-characterized drugs in each hepatocyte system, and then compare our data to the literature. Our ideal drug would be low clearance with clinically determined FMs and at least 2 routes of CYP metabolism. As I've said, we were sort of limited by the selection of well-characterized low clearance drugs in the literature. And whilst our first case study drug, diazepam, fitted all the criteria, our second drug, repaglinide, didn't fit the low clearance category. It did meet all the other criteria, however, and was deemed suitable for this exercise. And Chuck will discuss our case studies later. Next slide, please. So this slide shows the hepatocyte systems that we chose to evaluate. We realized that other systems are available, but we wanted to focus on those with literature evidence for their usefulness in determination of low clearance metabolism, and systems that had at least some partial characterization for this role. So we didn't want anything really brand new. We chose 2 plated 2D co-culture systems, HuREL and HepatoPac, and the 3D spheroid system. All of these are long-term systems. Our fourth system was the hepatocyte relay system. This is a hepatocyte suspension system which requires transfer of supernatant between fresh wells every 6 hours. So it's a short-term system, but one that's been used for reaction phenotyping of low clearance compounds in the past. One important point that I want to make from this slide is that the donor pools varied between each of the hepatocyte systems. So the donor number differed quite substantially, ranging from 1 in the spheroid system to 13 in the relay. And then donor age was also a major difference. For example, infants were included in the relay pool, and older children in the HuREL pool. And then in both HuREL and HepatoPac, there were geriatric donors. And donor ethnicity was also another variable between the systems. And collectively, these differences may have contributed to some of the intersystem variability we saw in our results through demographic effects on expression of CYPs or potentially other DMEs, for example, age-related differences, presence of polymorphisms. And this is just something to bear in mind while Chuck's discussing our case studies later in the presentation. Next slide, please. Okay, so I'm going to hand over to Ting now, who's going to talk about our inhibitor evaluation.
Thank you, Helen. I will from here I will cover the inhibitor evaluation in the selective models. Next slide, please. Next slide. Thank you. So as Helen mentioned, for inhibitor selection, we first go through the literature research, and we exhausted the literature to evaluate almost like 100 inhibitors. Among them, 75 were identified like CYP inhibitors. For the inhibition mechanism, we prefer the mechanism-based inactivation over reversible inhibitor. Also, the metabolic stability and the potency and the selectivity for the target enzyme are most important factors to consider. Additionally, because we are using long-term culture system, so induction potential and the cytotoxicity during the incubation length are also the factors to consider. Collectively, from the literature research, we came up a list of inhibitor candidates. Then the next step is the preliminary test by doing the first stability test in the HLM. And we use two concentrations. One is around the literature reported Ki, and another is like 5 times Ki in general. After this first screening, then we use the HepatoPac and HuREL incubation to test the selected inhibitor and the probe substrate pair. Here we focus on the inhibition extent, potential cell toxicity, parent compounds, stromal cell stability, because for HepatoPac and HuREL, there's stromal cells. We just try to avoid the background activity. And the inhibitor replenishment rate, again, because those three for HepatoPac, HuREL, and the Spheroid as long-term culture. So the inhibitor replenishment is also a consideration for the experimental design. In combined with the PXR, we came up a refined inhibitor conditions and to proceed with the formal assay. Next slide, please. This is the table. From left is the final list of the inhibitor candidates, and from left to right is the PXR EC50. As we can see, other than the benzophenobarbital and azamulan, other inhibitors showing like the EC50 greater than 100 micromolar. And as the HLM stability, only cannabinic acid showing less than 10% of the parent remaining at the end of incubation, which was a 2-hour incubation. However, interestingly, in HepatoPac stability, cannabinic acid, not like in HLM, are pretty stable. With this experiment result, we designed the replenishment date, the schedule. So for viability at the end of the incubation, we measure or test the cell viability. As we can see, other than pyroxytine, other compounds show, or other inhibitor and the substrate pair shows no effect on viability. However, the original selection of the pyroxytine concentration, which is 25 micromolar, showing the hepatocyte and the HuREL in the hepatocyte in both HepatoPac and HuREL detachment. Based on the observation, we decrease the pyroxytine concentration in the formal assay. Next slide, please. So this is the final study design. On the top part is the 3 long culture systems, the HuREL, HepatoPac, and the Spheroid. So for prepare the system, all three systems the hepatocyte need to be plated 6 days at least prior the initiation of the assay. And on day 0, for all three systems, we incubate the inhibitors with the hepatocyte for 24 hours. And after 24 hours, all the cell medium with inhibitor were taken out and replenished with either substrate or substrate with inhibitor. From here, every other 24 hours, we take time point till the end of the incubation. And also, we also take the T0 and 15 minutes right after the addition of the substrate in case that there is a significant non-CYP binding. For relay, because this is a suspension hepatocyte, the design is different than the long culture system. Before, on the day of the experiment, the inhibitor incubate with the hepatocyte for 30 minutes, and after 30 minutes, the medium containing inhibitor was taken out, and only the substrate added after the for incubation. And from here, the time point was taken at 4, 8, 12, 16, and 20 hours. Next slide, please. Here we're showing a representative example of the result. On the top is the 3A alprazolam with erythromycin parent depletion. We plot the line of the parent remaining against the assay time. Here we test like 2 concentrations in general for each inhibitor, a high concentration and a low concentration, span around like 10-fold in between. The blue line is the no inhibitor, the parent depletion without an inhibitor, and the red line in general is with the lower concentration of inhibitor, and this green line is with the high concentration of inhibitor. The bottom part is the metabolite formation, still follow the same conditions. And we use the linear phase of the metabolite formation to obtain the metabolite formation rate, and use those metabolite formation rate to determine the inhibitor potency at different concentrations. The reason we use metabolite formation rate instead of the depletion rate to evaluate the inhibitor inhibition potency is because those metabolites monitored are the CYP isoform specific metabolite. And also the other thing I want to point out is with the later time point, for example, on the bottom left, in the HepatoPac incubation, there's up and down. The down phase for the metabolite formation is the downstream metabolism of the primary metabolite. Next slide, please. So this is another example of 2C19 using the benzophenylbarbital as an inhibitor. What we want to mention here is that for this inhibitor, measuring by the metabolite formation, this inhibitor did show excellent inhibition potential for all the systems. Next slide, please. So this is an overall data summary of all the inhibitors behavior in all the 4 systems. Here we use the 80% inhibition effect as a threshold and the dashed line on the plot showing the 80% threshold. As we can see, for almost all the system, even at the low concentration of inhibitor test, already exhibit the over 80% inhibition effect. Next slide, please. So this is a table to show that the selective inhibitor conditions for the next FM determination was highlighted here is the difference of the inhibitor conditions between the 3 long-term culture system and the relay. So the difference in those selected conditions due to the different lengths of the inhibitor incubation time in various hepatocyte models. And with that, I will hand to Chuck for the next session. Thank you.
Great. Thank you, Ting, and hello, everyone. Thanks to Helen and Ting for describing the study designs and how the working group here selected the inhibitor conditions for these 4 hepatocyte systems. Can we go ahead and advance slide? There we go. And so now to further test the ability of these conditions of the irreversible chemical inhibitors to calculate fraction metabolized for CYP enzymes, we selected 2 drugs known to be metabolized by CYP3A4 because that's a major pathway, plus 1 or more other CYPs. And for these FM studies, then we needed to test all 8 of the CYP inhibitors in all 4 hepatocyte systems. And as Helen mentioned earlier, we were very methodical, I think, in trying to select the criteria to determine what our case study drugs would be. We wanted drugs that were extensively metabolized and well characterized in vivo. We thought it would be valuable to compare our results to previously published FM estimates that were determined in some of these same hepatocyte systems. And we wanted to be able to track the metabolite formation to help us confirm the inhibited pathways, though we did not purchase all of the secondary and tertiary metabolites that are known for the selected drugs. So with all these criteria, diazepam and repaglinide were really the only 2 that checked most of the boxes here, with the exception that repaglinide we know is not a low clearance drug, but it was actually the only other one that had been, I would say, fully phenotyped across the major CYP enzymes in a long-term co-culture system. So it's an important comparator for us. Some potential complications, though, with these 2 drugs are that they both do show multiple downstream metabolites that, in these types of in vitro systems might accumulate because they're basically closed systems. So it's something to keep in mind. Next slide. So we'll start with diazepam and a brief overview of the disposition of diazepam. It's a low clearance drug in vivo and in most in vitro systems. It's completely metabolized in vivo and then subsequently eliminated as glucuronides. As shown in this diagram, there's generally recognized 2 major CYP pathways. 2C19 makes the majority of nordiazepam, and 3A4 makes the majority of temazepam. Although there are minor pathways, 2B11 and 2C9 are known to contribute to the nordiazepam pathway. Next slide. So these are the results of our studies with diazepam and all of the inhibitors. The table here on the left is showing inhibition of parent depletion as determined from the intrinsic clearance values. And for the CYP3A4 FM estimates, even though we looked at 2 different inhibitors, we used erythromycin because it gave superior inhibition of our positive control alprazolam, and it also just appeared to give more consistent results across the different hepatocyte systems. So if advance to show the blue box on the slide, yeah, thanks. You can see all 4 systems detected the 3A4 contribution. That's shown with the erythromycin row down there. While only the relay system appeared to detect 2C19 metabolism with N-benzylphenobarbital. With benzylphenobarbital, actually the rate of metabolism surprisingly was increased in the long-term cultures, HepatoPac, HuREL, and spheroids, as shown by the arrows in the table. And if you advance the slide to highlight some different things here. One more. Yeah, interesting, and maybe a promising observation here is that for the minor pathways, where we know 2B6 and 2C9 are involved in metabolism of this drug, 3 out of 4 of the systems actually were suggesting some metabolism through those pathways. So that's good to see. So these same numbers now in this table, focusing on 2C19 and 3A4, were replotted into the figure on the right. And so you can see that we're comparing now these FM values to the known in vivo FM values. And those known values were taken from both DDI studies and 2C19 poor metabolizer studies. And so where our systems were able to allow us to calculate FM, it appears that our values did agree with the in vivo data. Although in the case of 3A4, you can see that that range is rather broad. Next slide. So we thought you'd be able to actually you would like to see the actual the curves here from these studies. And so this is diazepam again, with and without CYP inhibitors. The control with no inhibitor in these plots is shown in the blue color. And you could see that in the magenta colored lines in the long-term hepatocyte systems, the depletion rate is increased. As we saw in the previous slide, it looks like we were getting more metabolism with that 2C19 inhibitor, benzylphenobarbital. And in addition to confirm what we were seeing here, using HepatoPac as an example, in the 2 plots on the lower half of the slide, the metabolites formations of nordiazepam and temazepam were also increased versus the control. And so we think this is CYP induction effect because it was only found in the long-term incubations, and because as Ting described, we knew that this 2C19 inhibitor is a PXR activator, although there's almost nothing published on that. So I think that's interesting. Furthermore, there's a group at Merck that observed a similar effect in HepatoPac in their long-term incubations with a different phenobarbital analog that's commonly used as a 2C19 inhibitor. So a little disappointing that we were getting this induction, but it's also an interesting finding because it suggests these phenobarbital analogs are not going to be suitable for these types of studies. Next slide. And just to remind you, Ting showed this earlier, but benzylphenobarbital actually looked great in our pilot studies because clearly it was good at inhibiting the metabolism of the 2C19 substrate mephenatone. And the effects were also a little surprising because the concentration we selected of benzylphenobarbital was 6-fold lower than the EC50 for PXR activation. So I think what this means is that we think we're seeing induction, but we're probably inducing enzymes that aren't 2C19, and that's why we didn't expect to observe these findings. All right, next slide to show you the metabolite formation data with temazepam. With diazepam. We're focusing on just temazepam because these studies generated a lot of data, and to make this more digestible, what we've done is plot the 3A4 metabolite formation to temazepam by the relative changes. Okay, so these plots show the relative change in metabolite formation. And so if there was inhibition of the metabolite formation, then the data points would lie to the right of the dotted line. But if there's an increase in the metabolite formation rate, you would see the data points fall on the left side of the dotted line. And if you advance to show the boxes on the slide, can you hit advance, Cassie? And then thanks. And then, so as expected, the 3A4 inhibitors, decrease formation of the 3A4 metabolite. So no surprise there. That's good. And if you advance again, there were some metabolites that increased. Okay, so gemfibrozole glucuronide, or 2C8 inhibitor, increased metabolite formation. We speculate this could be due to UGT inhibition, since this compound is known to accumulate in hepatocytes. That's one possible explanation we haven't confirmed yet. If you peruse through the rest of this slide, you'll see some other cases where metabolite increased. This might be due to things like metabolic switching, or maybe changes in sequential metabolism that are occurring in the long-term incubations. But at this point, we're not going to speculate further. I think to really nail down all of and explain all of these findings, we'd need more extensive metabolite profiling. And as I mentioned earlier, we did not monitor all the secondary and tertiary pathways. Okay, next slide. To conclude, we also performed our comparison for our diazepam results with some published literature values. We found good agreement with the relay system to previous results, despite even using different inhibitors than what was used previously. So that was nice. For HepatoPac, it was curious that while we found evidence of CYP induction with benzylphenobarbital, the published data demonstrated 2C19 inhibition, specifically with diazepam. So that's an interesting difference here that requires a closer look. Next slide. So 3A4 fraction metabolized, it's consistent with what we know from in vivo data and even literature hepatocyte values. But obviously the long-term incubation systems were not amenable to characterizing 2C19 with gemfibrozole glucuronide. Okay, let's go to the next one. Our second case study drug was repaglinide, another extensively metabolized drug. In this figure near the top, M4 is the major 2C8 mediated metabolite. And then on the bottom in the blue box, M2 is generally thought to be the major 3A4 metabolite. As we pointed out, repaglinide is not a low clearance drug, but we thought it'd be a valuable case study because there are conflicting reports on the 2C8 metabolite M4 in DDI studies. In fact, during our investigations, this working group, we dug pretty deep, and we actually found that the 2C8 metabolite M4 was incorrectly assigned as being 3' hydroxyrepaglinide. And so thanks to Angela's team at Pfizer, the actual 2C8 metabolite was confirmed to be 4' hydroxyrepaglinide, as shown in the publication recently in DMD. If you advance once, we'll see the reference pop up. So, maybe this finding explains some of the controversy here with this metabolite. Like, is it possible that some people have actually been quantitating the wrong metabolite? pretty interesting finding. Maybe time will tell there. But we also want to acknowledge the Pfizer group who scaled up the actual metabolite for us so we could use it to quantify the 2C8 activity in our studies. Okay, next. Here's the result table for repaglinide. And most of the hepatocyte system did detect both 2C8 and 3A4 contributions to metabolism. So qualitatively, this is what we were expecting. But you can see when we replot this in the figure on the right and compare it to the in vivo estimates that our values are on average a bit lower. The total FM values are also all less than 1, at the bottom of the table there. So possible explanation for this could be greater than expected contribution of glucuronidation. Again, we're speculating here, but I want to be clear that we didn't think that this was due to inadequate inhibition based on what Ting showed you and how our inhibitor conditions were actually quite effective at suppressing the various pathways we're trying to monitor here. Okay, next slide. And this is the 2C8 metabolite formation. Again, inhibition will cause the data points to shift to the right and activation to the left. There's clearly very good inhibition of 2C8 using gemfibrozil glucuronide. Interesting, if you look down at the 3A4 inhibitors at the bottom, kind of looks like there might be some metabolic switching, meaning that the 3A4 inhibitor is shifting metabolism back to 2C8. As was observed with diazepam, there's a few other cases here where metabolite formation rates increased. So again, that would probably require some more extensive profiling of metabolites to fully make sense of all of that. Next slide. For repaglinide, there was a similar study conducted in HepatoPac, and our results agree with that earlier study on the contribution of CYP3A4. If you compare these, the bolded FM values. But our 2C8 inhibitor condition suggested an FM2C8 value that was lower than what was reported in that other paper. And that other paper used a reversible inhibitor, montelukast. That's probably the only reason why we didn't use the same inhibitor, because as we described, we really wanted to focus on using irreversible inhibitors. So this is possibly an important data point because it forces us to question whether we're missing something by using gemfibrozil glucuronide, or is it possible that the other inhibitor, montelukast, is maybe it's not as selective in some of these hepatocyte systems, and maybe that's why they got a higher FM value. But not sure. But I think it's something, again, that deserves some a closer look. Next. In summary, most of the hepatocyte systems did identify the two major CYP pathways of repaglinide, but with lower FM values for 2C8. And that concludes the data discussion. We'll go ahead and advance the slide, and we will next, though, provide final conclusions for all these studies. So in summary, several hepatocyte systems are now available, and I think we're all using them. And they've increased our ability to measure intrinsic clearance of low turnover compounds. Yet determining FM can still be challenging, right? So we spent years carefully selecting and testing different CYP inhibitors and found many potentially confounding issues with them. Even when we were convinced we had really selective inhibitor concentrations, we were dealt some of these unexpected results. But I also think that's part of the value in the data we generated here. And I think it suggests that we need more research in this area, and we're just kind of scraping the surface by looking only at SIPs as well. So there's a lot more to be done. Our studies, I think, highlight that as long as you can measure depletion of a low turnover compound, FM determination's going to be possible. That might not be the hardest challenge we're facing. I think conversely, if you have a drug that shows no measurable depletion, so really low turnover, and has multiple enzymatic pathways, I think we're going to need to characterize the complete in vitro disposition and really know all the metabolites, because these are static systems. And if you build up certain metabolites, they may be doing other things. They're going to change your results. Bottom line, we think all of these hepatocyte systems can be useful and are even promising for determining fraction metabolized by cytochrome P450. But our guidance would be that, for those of you who want to select inhibitors, just be careful characterizing them and qualifying them in your systems, because there's numerous reasons why your results may vary from published data due to the inherent system differences and long incubation times. And with that, I'm going to pass it back to Angela for Q&A. So thanks for your attention.
Thank you, Chuck. Thank you, Helen and Ting as well. We very much appreciate the work and the time that was put into making the slides into the presentation. So I think at this point, we will open it up for questions. And we do have a couple of questions online. I'll open this one up, and perhaps Ting or Chuck would be interested. Whether this is from Elaine Seng or Scott Obach is unclear, but it comes from Pfizer, just so you know. There's more work needed on chemical inhibitors. Full curves are needed. The inhibitors should be monitored during the incubation, and a full metabolite profile of the substrate should be monitored. Thoughts on that? I think there are two topics here. One is how well the inhibitor should be characterized, and two, the metabolite formation approach versus parent depletion approach. Anybody want to tackle one of those?
I can go first to just give my thought. First of all, during the incubation, we also monitor the inhibitor concentrations over the whole incubation course. That for sure, like at least if that's a concern. On the other hand, for metabolite, regarding for metabolite formation or parent depletion, I would say if we know all the information for the metabolite formation, it would be great to understand or to monitor the metabolite formation. However, I would say depend on the stage of the compound development, especially at the very early stage of the compound development, not like those probe substrate. We know the isoform specific metabolite, but at the very early stage when we don't know a lot of information about those metabolites, probably monitoring the parent depletion will give us overall contribution of those enzymes, especially now we establish the system with those inhibitor concentrations that understand the concentration we use now is or can inhibit those enzymes to pretty much like 90% or over 90%.
And I have nothing more to add. That was really good, Ting. And yeah, thanks for pointing out. We did monitor the inhibitor concentration throughout. Yep.
I think I would add, I think you did a very comprehensive answer, Ting. The only thing I would add is that a lot of these inhibitors that we're using have initially been characterized in microsomes as inhibitors. And so without really understanding what's happening to them in the hepatocyte systems, they require almost a complete additional evaluation in hepatocytes to make sure that they achieve the same level of selectivity and inhibition. So that's additional work that would need to be done that we certainly didn't have time to tackle here. Thank you. One more question here. Would you consider these systems and experiments as sufficiently validated with all their caveats to support FM data for regulatory submission? You want to tackle that one, Chuck?
Well, I would just I just kind of go back to I know we weren't able to show all of the inhibitors versus established CYP probes, but those initial studies, I mean, I guess it goes to show you can really show for compounds that are well understood, the results look fantastic. And then when we tried to move to also dirtier like substrates where they're not metabolized by just a single pathway, a lot of unexpected things popped up. So I think right now our groups would say we think our initial conditions are a really good starting point, and maybe even some of our inhibitors and their concentrations and all the conditions that go along with them are actually suitable. But I think that as we've talked about all these different variables that are coming into play here, I think you'd want to generate all the data that would make you comfortable in answering that question.
I agree. More work to be done there. Another question. Some hepatocyte models were not shown in the metabolism for certain CYPs. Have you checked CYP expression in these models? We did not confirm active. I think I'll tackle this one and anyone who wants to step in. I think if I understand the question, we did not. Well, I guess you could say we checked the CYP expression empirically in all the models when we did the inhibitor evaluation. So in other words, we were able to determine the probe activities appropriately, that the metabolites of the probes were formed, and then we were able to inhibit them. We did not compare the activity of CYP expression across all these models. But we know that for many of the probes and for diazepam and repaglinide, their overall intrinsic clearance values by depletion were quite variable. So we expect that there are differences in CYP expression and activity across the models where our data has shown that. I don't think we showed the intrinsic clearance values per se in the presentation, but they do vary across the models.
Yes, thank you, Angela. Can I just add
Yeah.
one point? It's that just like Angela said, we did see obviously isoform specific metabolite formation for all the probe substrate tested. On the other hand, it's that the inhibitor evaluation for this project is other than how we want to establish the inhibitor conditions for as many models as possible to expand the tool in the toolbox. Another important thing, like at least to me, is have this inter-system comparison. Is that we establish, let's say, for example, HepatoPac, right? We understand the inhibitor concentrations in the HepatoPac system itself for future, application. So in one hand is I feel that just or compare the protein expression across all the system is a little bit like out of the scope of the study. It just get myself thinking.
Thanks, Ting. Maybe we'll take one more question here. This one is, was the CYP isoform activity determined to be constant throughout the duration of the cultured systems? For example, does day 1 and day 5 activity match for a standard probe? I'm not sure. Ting, if you want to tackle that one.
Yes. For the activity, we did see the linear depletion of the substrate, the probe substrate, most of them from day 1 to day 7 for the long culture system. Of course, the HepatoPac and HuREL, because spheroid only and extended to 80 hours. So from the linear depletion standpoint, I would say yes, we are pretty confident that the hepatocyte function well through the whole culture's duration, and plus there are numerous literature data of those two systems are pretty consistent that the system can be well maintained and functioning well, consistent in 7 days. Thank you.
Thanks, Ting. I think we're at time here. Again, I just wanted to thank everyone for attending. And certainly let us know if you have any additional questions. I'll pass it over to you, Cassie.
Yes, thank you everyone for joining. You can view a recording of this webinar. Will be posted on our IQ YouTube channel. If you have any additional questions, you can submit them through the IQ Secretariat. Thank you, everyone. Have a good day.